So last class on embryology was the first week development. So in this session we'll be dealing with the second week development. So second week development, main thing is the process of implantation started in first week gets completed. So we'll study under three headings like changes in trophoblast, changes in embryoblast, and changes in endometrium, and that too will go day wise. So in day 8, what are the changes in trophoblast? So trophoblast will divide into two parts, inner cytotrophoblast. This divides to form the outer syncytial trophoblast. So syncytial trophoblast produces human chorionic gonadotropin. So again embryoblast will divide into low cuboidal cells towards blastocoele, which is the first germ layer to form. This is the hypoblast. And then columnar cells towards the endometrium, that is epiblast. So a cavity forms within epiblast called amniotic cavity. The cells of the epiblast lining the roof of amniotic cavity are the amnioblast or the amnion. So thus the bilaminar germ disc is formed. So bilaminar, two layers, hypoblast and epiblast. So these are the two layers of the germ disc. So let us see all these points in the next picture. See here, there is a bilaminar germ disc, epiblast and hypoblast. So hypoblast is towards the blastocyst cavity and epiblast is towards the ectoderm, and you can see the formation of the amniotic cavity within the epiblast, and here you can see this is the amnioblast lining the amniotic cavity, the roof of the amniotic cavity. And here you can see this is the cytotrophoblast, and this is dividing to form the syncytiotrophoblast. The same thing which is observed in a histological section. The epiblast, columnar cells, the hypoblast, low cuboidal or flat cells. You can see the amniotic cavity. You can see the syncytiotrophoblast with lacunae. So day 9, what are the changes in trophoblast? So blastocyst further penetrates endometrium. The site of penetration is closed by fibrin coagulum. Syncytiotrophoblast forms multinucleated mass with several small spaces called lacunar spaces, which coalesce and become larger spaces. This is the lacunar stage. So what is happening? So further penetration, and there is appearance of spaces within the syncytiotrophoblast which coalesce to form larger spaces, lacunar stage. Embryoblast, there is formation of Hughes membrane or exocoelomic membrane. So it is the flattened cells from hypoblast line the inner aspect of the cytotrophoblast forming Hughes membrane. Thus lines the primitive or primary yolk sac or the exocoelomic cavity. So now the bilaminar germ disc lies between amniotic cavity above and primary yolk sac below. Let us see here. So this is the hypoblast. It will give rise to the flattened cells which line the inside of the cytotrophoblast and forms the Hughes membrane. So this is the Hughes membrane or the exocoelomic membrane. And this is the exocoelomic cavity or the primitive yolk sac. Okay, and you can see here within the syncytiotrophoblast, the lacunae are appearing and fusing to form the larger spaces, lacunar stage. And here you can see the fibrin coagulum closing it. So next, day 11 to day 12. So what are the changes in the trophoblast? The site of implantation is completely healed. That means there will be complete lining of the endometrium there, epithelial lining will be seen. The lacunar spaces continue to enlarge and the syncytiotrophoblast forms trabeculae. The maternal blood vessels open into these lacunar spaces and there will be establishment of uteroplacental circulation. More prominent changes in syncytiotrophoblast are observed in embryonic pole, where the future placenta will be formed. So what are the changes in embryoblast? So first thing, formation of extraembryonic mesoderm. So earlier I have shown that Hughes membrane. So between the Hughes membrane and the cytotrophoblast, there will be formation of extraembryonic mesoderm, and then formation of chorionic cavity. So within this extraembryonic mesoderm, there will be formation of small cavities which coalesce to form the extraembryonic coelom. So then we formation of connecting stalk. So with the formation of the extraembryonic coelom, you can see the entire amniotic cavity and yolk sac will be separated from the cytotrophoblast, and there will be only small connection, that will be the connecting stalk. I'll show you in the next diagrams, which develop into the umbilical cord. So extraembryonic mesoderm is having two parts. The somatopleuric layer which lines the cytotrophoblast and amnion, whereas planktopleuric mesoderm is the one which is lining the yolk sac. So embryotrope is a fluid which is present in lacunar spaces which nourishes bilaminar germ disc before establishment of uteroplacental circulation along with the yolk sac. So yolk sac is the primary nutrition. So this embryotrope also helps in some part of nutrition. So then the chorion is the one which is the somatopleuric mesoderm of the extraembryonic mesoderm and the cytotrophoblast. Both together form the chorion. So amnion was formed by amnioblast, whereas chorion is formed by somatopluric part of the extraembryonic mesoderm and the cytotrophoblast. And the syncytial both trophoblast and the somatopluric extraembryonic mesoderm forms the chorion. And the cavity lined by the chorion is now called chorionic cavity or the extraembryonic cavity. You can see here, this is the exocoelomic cavity, and here they are forming the extraembryonic coelom or extraembryonic cavity. And this particular part with the trophoblast and the somatopluric layer of the extraembryonic mesoderm, that forms the chorion. So here is the amnion, here is the chorion. And this particular part of the extraembryonic mesoderm, which is lining the primary yolk sac, that is the splanchnic part of the extraembryonic mesoderm, whereas the part of the mesoderm which is lining the trophoblast as well as the amnion, this entire thing is the somatopluric layer of the extraembryonic mesoderm. So between these splanchnic layer and somatopluric layer, so there are formation of small cavities, and ultimately the goal is to form the extraembryonic coelom. I'll show you the connecting stack in further slides. Now you can see day 13 to 14, syncytial trophoblast forms villi, and the cytotrophoblast will invade this syncytial trophoblast and they form the primary stem villus. So outer syncytial trophoblast and inner cytotrophoblast. Secondary and tertiary villi will develop in subsequent weeks. I'll show you the difference. You can see here the syncytial trophoblast invaded by cytotrophoblast. So this is primary villus. So when this will be invaded by mesodermal core, then it becomes a secondary villus. When there is formation of the blood vessels, then this is called as tertiary villus. So only trophoblast is primary, with extraembryonic mesoderm is secondary, with mesoderm and blood vessels is tertiary villus. So these two will be seen in lateral weeks of development, but here in the second week it is the primary villus. So changes in embryoblast, formation of the secondary yolk sac. So what happens with the formation of the extraembryonic cavity or extraembryonic coelom? A small portion of the primary yolk sac will be pinched off, and the primary yolk sac will reduce in size, and later on this hypoblast will produce cells which line the inside of the Husser's membrane. That layer of cells is the endoderm. So ultimately the primary yolk sac shrinks in size, and parts of it are pinched off as exocoelomic cysts, which are seen in chorionic cavity. The final smaller yolk sac lined by endoderm forms the secondary yolk sac. The hypoblast cells become columnar in small area near cranial end of the bilaminar germ disc, which forms the precaudal plate. The significance is, so it is the first sign of differentiation because this is formed in the head region. So that means the opposite of that will be the tail region. So that is the establishment of the cephalocaudal axis. And this precaudal plate acts as the organizer for head region. So you can see here, so there is huge formation of the extraembryonic cavity, extraembryonic coelom. So eventually this is separated from the trophoblast, and this cavity is the chorionic cavity. So here you can see the part of the yolk sac has been pinched off as the exocoelomic cyst, and here you can see completely the secondary yolk sac. So this is the bilaminar germ disc, and here you can see the hypoblast is being differentiated into columnar cells to form the precaudal plate. So endometrial changes overall in the second week. What are the endometrial changes? The endometrium has to change itself to allow the implantation easy. So blastocyst completely gets buried by 12th day. Site of implantation is completely healed. So blastocyst lies in stratum compactum of the endometrium, which is shed during the childbirth. So it is called as decidua. The part of the decidia that lies deep to the blastocyst is called decidia basalis, which is invaded by trophoblast to form the placenta. Whereas the other parts of decidia are decidia capsularis and decidia parietalis. You can see here. So this particular part will be the decidia basalis. Here there is a placenta formation, and the part which covers this yolk sac and this thing is the decidia capsularis, whereas the remaining endometrium is called as decidia parietalis. So decidia basalis, decidia capsularis, and decidia parietalis. So summary. So in second week, it is called as week of 2. So you should remember that trophoblast differentiates in 2 layers, cyto and syncytio. Embryoblast forms 2 layers, hypoblast and epiblast. Extraembryonic mesoderm will have 2 parts, somatopluric layer and subplanchnopluric layer, and there will be 2 cavities formation, amniotic cavity and yolk sac. So now if you see abnormal sites of implantation, here you can see within the abdominal cavity or the peritoneal cavity, and here you can see in the ampullary region. Here you can see in the the third one. Where is the third one? Third one. You can see it is the interior of the tube. Here it is this, even though ampulla is also a part of the fallopian tube. So this we call it as tubal implantation. It is far inside. Then this is the interstitial part. So fallopian tube have four parts: the infundibulum, the ampulla, the main muscular part, and the interstitial part. So now either of the place it can get implanted. So we call this as tubal pregnancy. And here you can see ovarian implantation. Here you can see close to the internal os. So this results in further formation of placenta previa. So all these are abnormal sites of implantation. So here you can see the ruptured ectopic pregnancy. So this is the tube. You can see the rupture in the tube. So this is the ruptured ectopic pregnancy. Most common complication of ectopic pregnancy, can be fatal also for the mother. You can see the embryo developing in the recto uterine pouch. Thank you.